and adhesion. Capillary action is when, turn this on too so it'll follow me. There we go. Okay, so when the water molecules, they're attracted to, they're hydrogen bonded to one another. I'm not drawing the OHs and the H's, but like in our lab activity with the construction paper, they, you know how the water in the beaker like this, and this is the level of the water, and the water moved up and it got wet up here. And that's called capillary action. It means that when the water molecules, they're attracted to one another, but they're also attracted to the molecules of the paper, and therefore move up, and that's why it can move up. If it wasn't attracted to the molecules of the paper, it eventually gravity would pull it down and so on. So they just call that phenomenon capillary action. Other questions? Yes?
On this section, I'm not sure how to say it. Amphipathic?
Amphipathic. This is when a molecule, and phospholipids are an example of this, which is in another section, but chapter 5. But remember, phospholipids had the head and the tail, and the head is polar and the tail regions are non-polar. And so this is what an amphipathic molecule means. It means it's a molecule that's part polar, part non-polar, okay? And so that's why, like in the cell membrane, we get the lipid bilayer, all right? Because the polar parts are facing towards the water and the non-polar parts are not. Other questions?
Number 17 of the water section.
Number 17. All right. All right. So characteristics of water resulting from hydrogen bonding. So let's just go and review the hydrogen bonding. So remember that water is a polar molecule because, so here's your water molecule. This is a polar covalent bond. The polar covalent bond means that oxygen is more electronegative, drawing the electrons closer to itself, and so as a partial positive charge, and these guys have a partial negative charge, and these guys have a partial positive. So the hydrogen bonding between water molecules is between the negative and the positive parts, and so this hydrogen bonding, this is your hydrogen bond, I'll just put H bond, results in lots of different properties. Basically all the properties that we talked about with water basically comes back to the fact that water is polar and is able to make hydrogen bonds with itself as well as with other things. Like the picture I just erased was with forming bonds with the construction paper in the water. So for instance, water cohesion and adhesion are a result of that, of hydrogen bonding, because they're hydrogen bonding with each other, that's cohesion, or adhesion if they're bonding with another molecule. Surface tension is created because of hydrogen bonds. High specific heat is created because of hydrogen bonds. Remember, high specific heat, it can absorb lots of energy before it heats up, and because of the water molecules being hydrogen bonded to one another, as they increase in kinetic energy, that hydrogen bond resists that, all right? And so therefore, that is an example. The high heat of vaporization, so it takes, before it becomes a vapor, it takes a lot of energy to break those hydrogen bonds, so it resists that. And so basically, all of the properties of water that we've talked about can go back to this hydrogen bond. Does that make sense?
Yeah.
Okay.
I just have a quick question on the format of this quiz, or test, sorry. Is it multiple choice or is it?
Yeah, so the format always, I model it after the AP test, which is part multiple choice, part written.
Okay.
And so that's what this will be. So how it'll be is I will give you the multiple choice first, and I always make enough copies so that you can write on the test. So if you're a person that likes to write on the test, cross things out, draw pictures, I'm a big picture person, I'll draw pictures, help me think through a problem or something like that. You can write right on the test. And you do the multiple choice part first. When you're done, you're gonna come up here and put your Scantron and test up here, where I'll have a pile of the written part, and you'll take that back to your desk, and then you'll do this.
Okay, thank you.
You're welcome.
Is it all in pen?
So the Scantron part you need to do in a pencil. But if you recall at the beginning of the year, I talked about how the AP test wants you to write in black pen on the essay portion. So I would like you to write in black pen to get used to that, so that you can get used to just crossing off your answer. If you change your mind, you can cross it off and just keep on writing. All right. And so there's plenty of room to write, and so on the paper that I give you. Blue pen is acceptable as well. All right, it is, but for some reason they prefer black pens. And so I always think, you know, these, like on the AP test, these people are grading, you want to make them as happy as possible like while they're grading your essay. So that's why if they suggest black pen, I suggest black pen. All right. If you write in blue pen, I'm not going to give you a 0. All right. All right. All right. Any other questions? Chapter 2.
Number 7 is explain the oxide rule.
Right, so remember that, like if you have an atom, here's the nucleus, and in the first energy level, there's 2 electrons, and then the next one, 8 can fit. So you kind of pair it up. So this would be full. That's the octet rule. And so if you have, let's say, an atom that has these 3 missing here, what that then tells you is that to have that full, that energy level full, it needs 1, 2, 3 electrons, and therefore can make 3 bonds. All right, so that's all you need to know for that. Other questions? On chapter 2 or 3. Go to chapter 4.
Can you talk about polypeptide bonds?
Yes, I will in chapter 5, because that has to do with 5. So give me a second. All right. All right, so let's go to chapter 4 then, if we don't have any more questions on 2 and 3. So chapter 4, there was only 4 questions referring to this. Let me just address here. The number 4 asked you to recognize the major functional groups, so that you just need to be able to do that. Locate them on a molecule, be able to know what characteristics they give a molecule, and so on. Also, number 3, going back to number 3, when it says to the 3 types of isomers, I am not going to ask you show me the geometric isomers, but you should be able to look at and know that molecules that have the geometric isomers are like this, where they're around a double bond, and let's do OH here. And so then you could have this where the OH is here, and OH here, H and H. These 2 are geometric isomers. I wouldn't ask you what kind of isomers they are, but you should be able to look at these 2 and tell me that they're isomers, okay? So that's what you need to be able to do, is just identify isomers, all right, by counting up the atoms. Remember, isomers have the same formula, different structure. So I wanted to clear those 2 things up, those 2 up. Chapter 4, yeah? In the functions.
Yeah, that's what I was
Yes.
to know. So if there's like
Yes.
a carbonyl, we good to know that does this to it.
Yes.
Do we have to be able to recognize the functional groups based on like the fact that it's alcohol, like based on like their names, or just
Based upon their names.
Yeah.
Yeah. Basically that whole chart that you did. Yeah. It's fair game.
The term uracil, it just isn't.
Okay, so we're gonna let's with chapter 5. So let's go with any more chapter 4 questions. Yeah.
What was uracil?
Is that okay? That's chapter 5. All right. So, all right.
What does like methyl look like?
the methyl group, it's CH3.
Oh, okay.
So that's the only non-polar functional group. All the other ones are polar. Any other chapter 4? All right, so now we're in chapter 5. So we have 2 questions, so we better get to answer that on uracil. All right, so uracil is a nitrogenous base. The nitrogenous bases make up, do you guys remember what macromolecule? Yeah, nucleic acids. So nucleic acids, I'm just gonna do a brief little overview of this real quickly here. You have DNA, and you have RNA, right? And both of these, DNA is a polymer of a nucleotide. And so the nucleotide, let's talk about nucleotides in terms of DNA. 3 parts to a nucleotide. It has a phosphate group, a pentose sugar, and the third one is a nitrogenous base. That's a nucleotide. RNA is also a polymer of nucleotides. I should say nucleotides, it's plural. And has a phosphate and a pentose sugar and a nitrogenous base. So what's the difference between these two? So this is where we get in, this is how it bleeds into uracil. So first of all, in DNA, the sugar is what? What's that pentose sugar here? It has to do with the name, why it's called DNA. Deoxyribose. Sugar over here with RNA is what? Is ribose. So that's one thing that makes it different because they're both polymers of nucleotides. The next thing is the nitrogenous base. The nitrogenous base for DNA, you have 4 different options that can be plugged in here. So all nucleotides for DNA have the phosphate, deoxyribose, and the thing that changes from one nucleotide to the other is the nitrogenous bases. We often abbreviate their bases by their first letter. So we have adenine, which is A, thymine, which is C, cytosine, T, cytosine is C, and guanine is G. So you could have any one of those 4 bases in that particular spot. Versus, what's different for RNA? Instead of T, there's U, which is uracil. So we have A, U, C, G. And so this part is different, the U and the T in DNA versus RNA, and that's uracil. So uracil is one of the nitrogenous bases used in the construction of RNA. So when we look at this then in making a DNA, let me draw a DNA molecule. So you have a phosphate group and the sugar, and let's say the base is A here. That's one nucleotide. So then we hook that nucleotide to the next nucleotide. So that's the bond between a nucleotide. And this sugar hooks to the phosphate group of the next nucleotide. And then there's the sugar, and let's say the base is C, and so on and so forth. And so then we have the next bond, and I'm not going to draw anymore here. And I'm just going to put dot, dot, dot here as a chain. I'm not done drawing DNA because what's true about DNA? There's 2 strands, it's double stranded. That's why they call it a double helix. And so on the other side, matching up with the A, I'm going to put dot, dot, dot here. That represents what? Hydrogen bond. This A is going to match up with a base of a nucleotide on this side here. What goes with A? What bonds with A? Just T. So the T here is bonded here, and this T is going to be hooked to a pentose sugar I'm gonna put the pentose sugar upside down like this, all right? And have the phosphate group like this. This is gonna be important later. It's called anti-parallel. They run in opposite directions. And so this phosphate then is gonna connect to the sugar of the next base. So here's your pentose sugar. And coming off of here, what base am I gonna have? G, all right, and so on and so forth. And I'll put dot, dot, dot here. And these guys are hydrogen bonded to one another. And so DNA is double stranded. It looks like a ladder. So this part right here, the sugar and the phosphate groups are called the backbone of the DNA. That's kind of like the sides of the ladder, and the bases are kind of like the steps of the ladder, the rungs of the ladder, hydrogen bonded to one another. One thing that I really can't draw is the helix part. So if I were to take this and twist it, all right, so it's not just like a ladder, it's like a spiral staircase almost, okay? And so that's DNA. If I look here for RNA, if I were to draw RNA, I'll draw it over here so it doesn't get mixed up with this. RNA is also the chain of nucleotides, phosphate, and then the sugar is ribose, and then let's say your base is U. And the same thing goes where the sugar is connected to the phosphate of the next one, and then you have the ribose, and let's say this is A, and we continue on and make this. This is a completed RNA strand. Big difference between DNA and RNA, in addition to the nucleotides, is that RNA is single-stranded versus double-stranded, all right? And uracil is part of that. Next,
Is RNA like a helix too?
no, it's straight, yep. We'll see when we do protein synthesis that RNA is only made, you make it from one side of the DNA molecule, that's why it's single-stranded. The other question, so I kind of just covered nucleic acids there in a nutshell.
You know how you said like the sides of the ladder with the backbones? Well, since RNA is single-stranded, what's the backbone of?
It's still, but only just has one backbone. It's still the phosphate sugar, and these guys are sticking out.
Oh,
Okay.
Polypeptide.
Okay, the polypeptides. So for those people looking at their review sheet, what, is that associated with a number, or no? Does that help? Kind of like maybe 15, or are you talking about how it's made?
Like what it is.
What it is? Okay. So polypeptide is a chain of amino acids, and so that's the basic kind of thought as far as building a protein. And so I'll just go over here. So if you hook one amino acid to another amino acid and so on, the order, that's called a polypeptide. This is constructed by putting together two amino acids and then the third one and so on and so forth. So do you want me to review how like they hook together? Like peptide bonds and stuff like that. So let's look at, let's look at these two amino acids and how we would hook those two together, all right? So if I were to make an amino acid has a carbon down the center, and in the center, and it makes 4 bonds. And so every amino acid has these 3 parts in common. It has the amino group and the carboxyl group. These guys are always across from one another. It has a hydrogen and it has an R group, and remember the R group is what varies from one amino acid to another. And so when we hook this amino acid to amino acid number 2, so I'm going to do the same thing for amino acid number 2. I'm just going to draw here the groups that make it an amino acid. So this is your 2 generic amino acids. And we want to hook them together. And so we always, whenever we're hooking the 2 molecules together, remember an OH is lost off of one and H is lost off of another. So these guys here lose a molecule of water. That's called dehydration reaction, right? And then this carbon is now going to bond to this nitrogen because this bond is no longer there and this bond is no longer there. So they bond together. That right there is called a peptide bond between the two amino acids. And so then if we want to build the next amino acid and add to that chain, we draw it again, and the amino group would lose an H and then OH and so on and so forth. And so you build your polypeptide. And so then remember that this chain of amino acids is what part of the overall construction of your protein. Remember there are 4 levels. This is the primary, all right? The primary is just the chain of amino acids. And then secondary would be that chain of amino acids with the alpha helix and the beta pleated sheets. Tertiary would be when you get the bends and folds like this, all right, and so on and so forth. And then quaternary would be if you have multiple polypeptide chains. Yes.
So for number 5, are there like a certain 4 characteristics of sugar that we need to know?
No, there is not really. All right, so sugar, I'll just give you a couple, all right, that are important. So sugars can vary in their construction based upon whether or not they're in water or not. So they're linear out of water and ring-shaped in water. Sugars are made out of 3 elements. What are the 3 elements that sugars? Carbon, hydrogen, and oxygen. So if you, like the nitrogenous bases here, for instance, they're in a ring shaped, but it's a nitrogenous base. So if you see nitrogen as part of the ring, a lot of people see ring and go, oh, that's a sugar. But if you see nitrogen in that ring, it's most likely one of the bases versus a sugar. So sugars are carbons, hydrogens, and oxygens. Monosaccharides, those are found in a unique ratio, 1 to 2 to 1, right, like glucose. And they're used for 2 purposes, used for fuel or energy, as well as a building structure, like cellulose, okay, like in the cell walls. Other questions?
For number 28, do we have to copy the table onto this paper, or should we?
No, you can just be able to do that. Yeah, yeah. 17? All right. So primary structure is the chain of amino acids. It's the order. So remember, there are 20 different amino acids. It's the specific order of those. And I do not ask you a question about how it may be deduced in the laboratory, so you can cross that. Okay. That was part of, I forgot to delete that off of the review. It was the part of the notes that I had you cross off.
Which question is that?
That was the second part of 17. Yes.
For number 8, it says distinguish between the glycosid and glycogens found in starch and cellulose, and then explain why the difference is biologically important. Can you explain the last part why it's important?
Why it's important? Because it changes the bonds between the glucose molecules. Starch and cellulose are both chains of glucose molecules, but remember there were 2 different forms. Starch is alpha and cellulose is beta glucose molecules. So the change in the bonds, the way that the bonds, I don't know if you remember that they alternate. You have to flip the glucose over every other one for cellulose. And so that right there makes it have a different structure, which then makes it a different function and makes it, cellulose is much stronger, harder to break apart than starch. And so I use the example for us when we eat cellulose, our digestive enzymes will not break apart cellulose, but will starch. And so that slight difference, makes, and so that's.
21, what is dehydration and explain how proteins can be denatured?
Okay, so denaturation is when the protein loses its 3-dimensional shape. So So remember that if you have a chain of amino acids, if a protein is linear and has the chain of amino acids, it still may be put together properly in terms of the chain of amino acids, but it's not going to do its function because it has to fold and stuff like that and have its three-dimensional shape. And so what happens is there are things that can cause proteins to lose that three-dimensional shape. Like this is the tertiary structure here, and like so this is stabilized by interactions between R groups and so on. And so things like changes in temperature, if the temperature gets too hot, the atoms of this molecule begin to increase in kinetic energy. These bonds between the R groups can break and the protein can kind of unravel and lose its shape. If it loses its shape, it can't do the right function. Other things besides the increase in temperature would be changes in pH and salt concentration are the main things that can cause it to do that. Other questions? Okay, so functions of nucleic acids. Oh, I erased my RNA. So DNA, I'll look at them as two different nucleic acids. So DNA, the function is to contain instructions to make proteins. And those instructions are in the order of the bases, the A's, the T's, the C's, and the G's. So they contain instructions to make proteins, which give you your traits. So that's the genetic traits that you have. So you have DNA to make the codes for making proteins that will get deposited in your eye to give you a certain eye color, to make enzymes in your digestive system, and so on. RNA, on the other hand, has a different function. There are different kinds of RNA. For right now, I just talked about like mRNA as an example, carries instructions from the DNA in the nucleus to the ribosome to make a protein. So it kind of like copies the instructions from DNA and takes it to the ribosome. So that's basically what.
Distinguish between a purine and
Okay, so that goes with the bases of DNA and RNA. Pyrimidine, there's two different, for the nitrogenous bases, they're a base which are ring shaped, but they have nitrogen. And some of them are a single ring and some of them are two rings fused together. And so the pyrimidines are one single ring and the purines are two rings fused together.
Number 11.
11.
Ester linkage.
Okay. Now, ester linkage is in the formation of a fat molecule. So general overview of a fat molecule, remember, has the glycerol as the backbone, 3 carbons, and you have to hook 3 fatty acids onto it. So you have a long chain of fatty acids here, and when we hook that fatty acid onto the glycerol molecule, dehydration occurs where you lose a water molecule, an OH and an H, and they hook together. And so that bond between the glycerol and the fatty acid is called an ester linkage. And so there would be 3 of those to make a full fat molecule because you have 3 fatty acids that attach here.
So that's formed by dehydration.
Yes. Yeah.
For 14, it says explain how amino acids can be grouped according to the physical and chemical properties of the side chains.
Okay. I just erased my amino acid. So remember your R group here. That's the part that changes between the amino acids. If that R group, this is your generic amino acid, if that R group was, let's say, a hydrocarbon. Our hydrocarbons, made up of just carbons and hydrogens, are they nonpolar or polar? Nonpolar. And so if that R group was made out of just carbons and hydrogens, we would call that group, it's a nonpolar R group, we would call that amino acid a nonpolar amino acid. So the amino acids are classified based upon the characteristic of their R group. So we have a group of amino acids that are nonpolar because their R groups are nonpolar. If, for instance, that R group instead of having hydrocarbons, let's say that R group had some functional groups, like it had a hydroxyl group and so on, which we know are polar. So then we can have molecule, the R group being polar, and we call that amino acid a polar amino acid. And then the last one is if it has a charge, like if the R group has a positive or negative charge, we call that electrically charged, and that would be your third type. So it's based upon looking specifically at the R group, the characteristics of the R group. But those are your three groups. Other questions?
Are the questions like 23 and 26, they're like really similar? One says like summarize the functions of nucleic acids, but the other one is list the functions of nucleotides. So like, how do you want us to do that?
So the nucleotide make up nucleic acids. So the nucleotide, the function of that is to hook together to either make DNA or RNA. Does that make sense? And then once DNA and RNA are made, then the DNA and the RNA have specific functions. Does that make sense?
Yeah.
Okay. All right. Questions? 18. Okay. Okay, the two secondary protein structures. So the primary was the chain of amino acids. So amino acid hooked to amino acid and so on. And so then your two secondary is the alpha helix. So this is where it kind of looks like that telephone cord. This is stabilized. There are hydrogen bonds between them and stabilized by hydrogen bonds between the different amino acids in this chain. So stabilized by hydrogen bonds. Whereas the double helix, or sorry, not the double helix, the beta pleated sheet. So here's the beta pleated sheet, looks like this. And oftentimes with beta pleated sheets, they curve around and there's another beta pleated sheet like this. And they hydrogen bond to one another like this. All right. And so with this drawing, I also included, because this curve actually is the tertiary structure when you get a curve here, but beta pleated sheets often are hydrogen bond to one another like that. And so you have, and that's the beta pleated sheet. And so stabilized by hydrogen bonds. And then once you get, so these two are the secondary. Tertiary I kind of drew in here as well, so you get this curving. And remember, that's different than these two because that curve, these guys are stabilized by the interaction between the R groups of, I mean, that's not part of that question, but I just threw that in there here to compare and contrast secondary versus tertiary. Tertiary, you get those curves due to the interaction of the R groups. Other questions? Okay, I'm going to go over the pattern matching activity answers. And then if you think of any questions as we're doing that, because that's also with chapter 5 as well, so we still have some time here. All right, let me erase this so I can, I'm just gonna display my answers over the projector. Okay, so group 1 was the amino acid. So it asks you to draw the amino acid up here, the generic amino acid showing all the bonds. So nitrogen makes 3 bonds with the hydrogen, and this is the amino group that has already acted as a base by accepting an H+ ion. And this is the carboxyl that has already acted as an acid by releasing its H+ ion, and that's why it has an O- here. So the oxygen that's part of the carboxyl group is negative, and that's the reason why. So both of these, this is still an amino group, this is still a carboxyl group, so we'll talk about that. All right, so the next one here asks you to draw the bonds between all the atoms of the carboxyl group. So we have the C double bonded to an O, and the C is also bonded to an O, which is bonded to an H. And it asks you for the next one, does the carbon atom bond the same way to both oxygen atoms? And that is no, because it bonds a double bond to one and a single bond to the other, and it can't double bond. It's going to bond to a larger molecule because it can only make 4 bonds. All right, so it can only single bond to that oxygen. All right, down here, it just gives you the idea that the R groups, which I circled here, change between one amino acid and another. So this R group is different than this R group. So this would be an example of that hydrophobic or nonpolar R group. So this would be a nonpolar amino acid. All right, versus this, see how this has functional groups on it? And so therefore this would be a polar amino acid because its R group is polar. So this goes back to the classification of those. So it asks you, and you could use, I didn't draw them because there's 20 different amino acids. You could have chose any of the 3 of the 20 amino acids as long as they had different R groups attached to them. And so when you had to identify them in your packet, so you should have 14 of them. And all of them have that carboxyl group, amino group, H, and then the R. And the way that they're written here is all the R groups are off the bottom on this one, all right? And so the way that they put this here, so those R groups, they give you a total of 14 amino acids, okay? So then it asks you what elements are found in all amino acids. All is the key word there, and that's carbon, hydrogen, oxygen, and nitrogen. Now, amino acids can have additional atoms or elements depending upon their R group, but that varies from amino acid to amino acid. So what's found in all amino acids, no matter its R group, are those 4. This one here is the formula is you take, there's 20 different amino acids available, and so if you want to see what's the combination or how many different combinations can I make if I just put 3 amino acids together, the X power is the length of the chain, so 20 to the 3rd power, so that would give you 8,000 different combinations in amino acid or polypeptide that's 3 amino acids long. So then you can see it gets much greater, so 20 to the 10th for 10 amino acids long, right? 1.024 times 10 to the 13th. If you could also move the decimal place over 13 times. They have a very large number, and then 20 to the 50 is 1.12 times 10 to the 65th. All right, so I think most people wrote it in this format. All right, so that is proteins. So that the purpose of this, I won't ask you a question to calculate this. The purpose of this is just to show you that you can get so many, you can make thousands and thousands and thousands of different proteins just from 20 by changing the length of them and also the different combinations in terms of the order in which they occur. All right, group 2 and 3 were about lipids. So group 2 is steroids. So this is cholesterol, and over here on this side here, it asks you to draw an arrow to point at the double bond. So right here is the double bond, and then it asks you to about how none of the hydrogen atoms have been drawn, and so you can fill in the missing hydrogen atoms. I just added them onto here. And so basically, how I knew where hydrogen was is that every point is a carbon atom. They didn't put the carbon atom, but every point is a carbon atom. And carbon can only make 4 bonds. And so I counted, like, for instance, this bond, this carbon here, already had 1 bond here and 1 bond here. It makes 2 more, so I added 2 hydrogen. And so that's how we did that. And then it asks you to paste your steroids. You should have 3 steroids here. They look like 4 fused rings. And by the same token that I just went over with the cholesterol on the opposite side, you could add the hydrogen atoms to the molecules. All right? And that is cholesterol. Only one with 4 fused rings. Fats are made out of fatty acids. And so looking at this here, it asks you down below, how many fatty acids do you have in your collection? There should be 3, and they look like this. 1, 2, 3. Fatty acids have a hydrocarbon chain and a carboxyl group. That's what you look for a fatty acid. So we have 3. Then the next question asks you, the next 2, how many are saturated? Saturated means no double bonds between the carbons. So these 2, what I put on the top and the bottom, have no double bonds. So we have 2 of those. And then monounsaturated, mono means 1, so that's this one here that has 1 double bond here between these 2 carbon atoms. So there's 1 of those. And so going back here, it asks you about what would polyunsaturated mean. It would have multiple double bonds. And it asks you to draw just a random short polyunsaturated fatty acid. So to be a fatty acid, you have to have the carboxyl group and a hydrocarbon chain. To be polyunsaturated, you have to have multiple double bonds. I chose 3 here. You don't have to have 3, you had 2. And so on, and I drew the appropriate amount of hydrogens to make the carbon have 4 bonds. All right, so yours doesn't have to look exactly like mine, but the same idea. Okay, the next group is the sugar group. So it asks you how many straight chain sugars do you have in your set? There should be 6 of those. All sugars you should have are carbon, hydrogen, and oxygen. There are 2 single rings and 2 disaccharides. So let's look at those on the other page here. So you paste them here. I'll come back up here in just a second. You have to paste them here. And so these ones here are your straight chain sugars. How do I know? They are made out of just carbon, hydrogen, and oxygen, and they have a double bonded oxygen somewhere in the molecule, and every other carbon has an OH, or hydroxyl group. And those are your straight chain sugars. Here are my 2 monosaccharides. They're a ring made out of just carbons, hydrogens, and oxygens, and these are my disaccharides, 2 of them put together, 2 rings put together. So then, for table sugar, you had to hook glucose with fructose. So it said to draw them below, so I drew them here. And to hook them together, you lose a water molecule. So an OH will come off of one, and an H will come off of another. All right, so to do that. All right. And then the last group is your nucleic acids. So nucleic acids, the nucleotide is made out of a base, a sugar, and phosphate group. So the first question is you identify the nitrogenous bases. There are 5 types of nitrogenous bases: A's, T's, C's, and G's, and we have a 5th one because RNA has U. All right, so therefore these are all of them. So notice here 3 of them are single rings, but notice that they contain nitrogen, so that's how you're going to tell that they're different than sugars. And then 2 of them are 2 rings fused together. So those are your 5 nitrogenous bases. Then building a nucleotide, we add the bases to the sugar. So here's your nitrogenous bases here in each one of these pictures. Here's a pentose sugar. Pentose sugar, and so on. So we should have 4 of those, nitrogenous base and the sugar molecule. And then lastly, it asks you to, now we're going to add the phosphate groups, and it asks you if there's 1, 2, or 3 phosphate groups attached to it. So here's 1, so base, sugar, phosphate group. So it's 2, base, sugar, base, sugar. And so nucleic acids, all what they have in common is carbon, hydrogen, oxygen, nitrogen, and phosphorus because of the phosphate group in the nucleotides. Alright? And the summary is just that. It's just a summary, the table. A lot of people had a lot of questions like, are we supposed to do something with this table? Because people were confused that there's blank spots. And no, see it says proteins, building blocks are amino acids, and then we talk about all the different functions of them. And so there's way more functions than class or building blocks, so they just left those blank here, and those are the different functions there. So you don't have to do anything with that, that's just an overview. I have a question.
I have a question. On the rings, like just in general, how would you write that out into a molecular formula?
Like these.
Yeah, like how would you? Yeah.
Okay. Like sugars or something like that. Or?
Yeah.
Okay, so they shorthand it. So when they shorthand it here, they usually like this ring here or one of these rings here. This one they put all the hydrogens and so on. This one they did not. So you can assume that at each point is a carbon atom, and then at each point you can see like this carbon right here would make this double bond here would make 2 bonds, and 1 here to make the 3rd bond. So it would have another hydrogen. Okay, so then you just count all the atoms.
Okay. I found myself like trying to go in the circle, like going in the circle, and I don't know where to start back. You know what I'm saying? Like, okay, so like each corner would be like a carbon atom, right? So that would be like CH. What would that be? CH2.
Yeah, but for the whole molecule, you're counting up all the carbon.
Oh.
You know what I mean? To get the
That's
whole
right.
formula. You're not doing it like parts of it. You know,
So then it would be like
count
C5.
all the carbons, count all of them, but see however many.
Oh.
Alright, so
Okay. Thank you.
yeah, no problem.